Hello. Okay. Mm-hmm. Mm-hmm. Okay. 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 Thanks. Uh-huh. So I'm so used to see Lucia from Skype now. Hello. Thank you, Lucia. Okay, so I would like to give a great thanks to the organizer first for inviting me and also for allowing me to be there remotely and for spending that time during the lunch break trying to make these things work. So I really appreciate that, and whenever you want to Zoom in Paris, I'll be there for you. Okay, so today I'm going to talk about the dynamics of the brain during reported and spontaneous conscious access. But first, I would like to set my general framework about consciousness. We've seen that consciousness is a very polysemous word, and we all put different, as scientists as well, different emphasis on different aspects. And the way I see the relationships between the different aspects of what we call spontaneously consciousness is the following, at least at the moment, after being in the field and teaching that field for a few years. I know some of you might disagree with the way I put things, but I wanted to show you how I see things now. And a key element of this framework is the idea that there is a basic mechanism that we call conscious access, a basic moment-to-moment mechanism that allows for the other more richer and more flourishing aspects of consciousness to emerge, including phenomenal experience, the building of a conscious self, etc. So today I'll be talking about investigating the dynamics of this little process here of conscious access. So what do I mean precisely by conscious access? I know many of you already know about that, but I can't resist presenting a demo of motion-induced blindness. Again, if you fixate in the middle here, the little blinking green spot, you will soon see that some of the yellow spots will disappear from your conscious access. They are still on the screen. They are still processed. They are still processed in your brain to some extent, but sometimes you have access to this information and sometimes not. okay? And that sets the problem very simply and very nicely. What's the difference in your brain when the same stimulus gains access and you have access to it and you can report it notably, and when you don't see it, okay? So that's the basic neurological question or neuroscientific question. And if we know the mechanisms that make this distinction in the healthy brain, we can also then try to go and interrogate what happens in the brain of patients with disorder of consciousness. Sandehan has spoken about these patients already. So patients for whom on the clinical examination, it's not sure whether they actually have conscious access to some information or not. Right, so the first study that we started a long time ago at the beginning of the 21st century with Sandehan to investigate the brain dynamics of conscious access used the attentional blink that has been mentioned, which is a really nice protocol where you can present a stimulus, for example, a word, and in exactly the same conditions. On some trials, you will tell me that you haven't seen stimulus, and on some trials you'll say, oh, yeah, I've seen it quite well, okay? And if we split the trials according to whether you've reported that you haven't seen the stimulus, can you still hear me? Yeah, okay. So if we split the trials according to your report and we look at the brain activity recorded with EEG evoked by the stimulus according to whether at the end of the trials you told me that you've seen or not seen the stimulus, we found a very striking pattern where for the first 300 milliseconds, the activity patterns were indistinguishable between trials where eventually you've seen the stimulus and trials where eventually you haven't seen the stimulus. And then suddenly we saw a divergence between these two with late sustained activity for seen trials and not for unseen trials. And if we reconstruct the cortical sources of these effects in the brain, you see here below seen trials and above unseen trials, we can see the film of the activations that occur in your brain when you become conscious of an information or not for exactly the same external stimulation. And that's how it goes. So you see that initially we have nice visual activations in the occipital cortex for both cases, and these activations move forward in the temporal cortex a bit more on the left because these were words, and with the same strength in both cases. And then you'll see that around 200 milliseconds after the presentation of the stimulus, something seems to have clicked on the trials where you'll subsequently say that you've seen and not on the other trials. And when you say that you've seen, you see a succession of activity with many brain areas involved, including frontal, parietal, but also you'll see that a crosstalk between frontal and posterior areas that do not occur when you say that you haven't seen the stimulus. Okay, so this validated the idea that there might be special dynamics associated with conscious access, and that it might correspond to an abrupt transition in the dynamics of the global brain response to the stimulus. And other really nice studies have been done around the same idea, using this time, for example, visual masking, where they saw, notably Antoine Delcu with Sandehan and also Sid Quider, where they saw that initial waveforms might show gradual increase with increasing strength of the stimulus, while later activity might show the discontinuous dynamics where these late activity is not evoked when the stimulus is below the consciousness threshold. Okay, so recently we thought, okay, if these very special dynamics are an important signature of consciousness, we must be able to use them in patients. But before we use them in patients, we must make sure that this is a valid general signature of conscious access. Why? Because we've seen these dynamics essentially in visual paradigms, and also with complicated visual paradigms. So the attentional blink, masking experiments with sharp transients in the stimulation itself. And another element of generality that we wanted to bring is relates to something that has been very much debated this morning, which is, okay, these late activity are essentially found in protocols where also you ask subjects to report the stimulus. Might they be only related to the tasks that they're doing? Okay, in which case it's not really a signature of conscious access, it's a signature of task processing. Okay, so we wanted to bring all this generality. So what I'm going to show you first is an experiment on healthy subjects, trying to see whether we can show the generality of this signature, this potential signature of conscious access. So what we did is build a protocol where we use audition instead of vision. And also practically it's very important to bring this protocol into audition if we want to be able to test disorder of consciousness patients, because it's much easier to have auditory stimulation for these patients. Also, we built a protocol where we have a much simpler stimulation. So stimulation around threshold, no masking, no fast transient in the stimulation, presumably a bit more ecological than what we've been using so far. And I'll explain exactly what stimuli we use. And lastly, we also wanted to test the same protocol with and without a task to see whether these dynamics hold when you don't have to make a task on the stimulus and you just spontaneously become conscious of the stimulus. Okay, so more precisely, so these are studies that are done with many collaborators, but especially I started them with a student called Ghislaine Labouré. So in this protocol, you're listening to a continuous noise like this. And within that noise, we insert vowels that can be A or E, the French ones, A or E, but the French A or E, presented at different intensity level relative to the noise, okay? So what you see here are negative dBs because the intensity of the stimulus was always a bit smaller than the noise, but you still hear them, at least for the strongest one, minus 5 dB, and the threshold for hearing the stimulus was, and you'll see that around minus 9 dB, okay? And on some trials, there were no sounds, okay? Very, very basic protocol. And each of our 20 participants took part in 2 types of sessions, an active session where they reported the identity of the vowel and the audibility of the vowel on a scale, and a passive session in which the presentation of the stimulus was exactly the same, the period that we recorded was the same, but instead of asking a report at the end of each trial, a report on the sound, people were instructed to ignore the sound but do a quiz questionnaire. For example, they will be asked, when did Notre Dame burn, for example? Oh no, that was before Notre Dame burnt, actually, but right. And okay, and so 2 different sessions, counterbalance across, the other was counterbalance across subjects, okay? And importantly, we wanted to have a more formal framework for analyzing the dynamics that we would observe in this experiment, and more explicitly state what we mean by discontinuous dynamics, bifurcation dynamics, okay? So that's why we made some simulation and predictions. So one first type of dynamic that we can encounter is what we might call unimodal dynamics. So when we increase the stimulation here, we would see neural activity increase with increasing mean of neural activity, and all the different trials will group around that mean so that we have an increase in the unimodal distribution of activity with increasing stimulation, okay? What we mean by bifurcation dynamics is a very different dynamics, whereby when you're at threshold, the same stimulation will, on some trials, group around a low mean, just like the baseline. So, in other words, this process has not been triggered, and on some trials, this process has been triggered, and it will group around another mean, higher mean. So, probably corresponding to either non-conscious and conscious processing, okay? So in this case, we predict not a gradual increase in unimodal distribution, but that around threshold, you'll have bimodal distributions of activity around 2 means, okay? Right, so the classical way to analyze neural data is to average activity across trials, okay? And but if you think about it, the implicit hypothesis behind that, the reason why we believe the mean is a meaningful summary statistics is that we imply that there is unimodal distribution behind that. But if we try to compare unimodal dynamics and bifurcation dynamics just based on the profile of averaged activity across trials, we see that it's not very diagnostic. We can find examples where the mean will be very similar in both cases, especially if the mapping between how we measure the stimulation in physical terms and its transformation in neural terms is nonlinear. We have no grasp on that, okay? But what might be really diagnostic is looking at the variability of neural activity across trials. And indeed, the predictions here for the variability are completely different for the unimodal dynamics and bifurcation dynamics. So variability for unimodal dynamics will show either a flat line or maybe some monotonous increase with stimulus strength, but certainly not a non-monotonous profile. Whereas for bifurcation dynamics, we predict a very peculiar profile where we should see a burst of variability across trials at threshold stimulation, because at that stimulation level, you have a split between 2 types of trials, okay? And while you go away from that threshold, either towards all trials are conscious or towards all trials are unconscious, then again, variability decreases because you have homogeneous population of trials around a single mean, okay? So what we did was to try and find in the neural activity we were recording whether we were seeing this prediction or this prediction at different moments after the presentation of the stimulus. Right, so first the behavioral results show that indeed our stimulation is around threshold and that the threshold is around -9 dB. That's the same result if we look at the audibility rating. And interestingly, with audibility rating, we can also see the variability of this rating across trials by the subjects. And what we see is this profile of burst of variability across trials when you're at threshold. And if you look at the distribution of the variability ratings, of the audibility ratings, we see that this indeed seems to correspond to a split between two types of trials. Trials where people say, I haven't heard the stimulus for exactly the same stimulus, and trials where they say, oh yeah, I've heard it quite well. So that matches what we observed a long time ago in the attentional blink, but we were surprised to see this really clearly also in a protocol that is much simpler with an auditory stimulus at threshold. Right. Another interesting aspect is that this peculiar variability profile in the behavior was found for each and every subject. And I guess that's important when you want to move to a single subject analysis then. Right, so now let's look at the dynamics in the recorded brain. So it's EEG. So in response to our simple auditory stimuli, so ARE, we found classical waveforms with a first auditory N1 and then a sequence of positivity that you can see here also on the topography in response to the best stimulus. So auditory N1 and then followed by a nice P300. Right, and so as I said, to analyze, to go deeper into the dynamics of these waveforms, we looked at the mean and variability profiles of these different waveforms. So as a first, we can do something really simple, look at the activity of a group of electrodes here that captures well these two important waveforms. And if we do that, we see that indeed activity increases in intensity with increasing stimulation. So that for the negative waveform and then for the positive waveform. And we notice that it seems to be nonlinear from very early period, around 100 milliseconds after the presentation of the stimulus. Now, what happens if we look at the variability across trials? Well, in the initial period, variability across trials seems to be flat or slightly increasing with increasing stimulation, but certainly not non-monotonous. But in the later phase, we clearly observe this very peculiar profile that was predicted by bifurcation dynamics. So with a burst of activity around our threshold stimulation values, suggesting that there might be a split between two types of trials when you're stimulating at threshold. Okay, so that was using a region of interest, but obviously now we can do much better than that and use the activity of all the electrodes to summarize the activity related to the presence of the stimulus using multivariate pattern classification. So if we do the same type of analysis with multivariate pattern classification, we see something similar. So for the average activity across trials, we see that there is a nonlinear increase from the very start, a nonlinear increase in activity with increasing stimulation. And if we look at the variability of cross trials, initially it's quite flat, and from 300 milliseconds onwards, it's, oh, okay, so you're, I wonder if you're missing the best graphics here. Yes? Ah, okay, right. So because I can't, but it doesn't matter. Okay, so so and we see that indeed, even if we take the whole activity of all the electrodes, we see that we this pattern of bursting variability at threshold stimulation is present for the global activity that we record with the EEG. Right. So the next question, now that we see that we validated this non-trivial prediction of bifurcation dynamics, the next question is, okay, if we have indeed a split between two types of trials, does this match the subjective report of the participants in the active session? So does it match their report that they've heard the stimulus versus not? So again, if we go back to our models, the predictions are different when you're thinking about the behavior of heard versus not heard trials. In unimodal dynamics, in the unimodal dynamics model, that really very much corresponds to standard signal detection theory models. You have a gradual increase in the unimodal distribution. You set a threshold above which you've heard, below which you haven't heard. And in this case, your prediction of the activity when you split the trials according to participants said that they've heard or not heard is the following. In both cases, heard or not heard, you should see an increase in the activity with increasing stimulation. Because, for example, if you're taking up the trials that fall below this threshold in this population, they are still going up with the general mean corresponding to this level of stimulation. So you have parallel increase in activity in both cases. For bimodal dynamics, we assume that subjects will set the threshold at the natural dynamics threshold of the brain, and so that when they don't hear the stimulus, it corresponds to trials where this neural event did not occur. So we're at the baseline for all the stimulation levels, and when they say that they've heard the stimulus, they are above this mean, and they might be there might be a gradual increase with increasing strength of the stimulus. Okay. No way. So I have to. So I have no, but you know that the time doesn't run the same way in Paris. Okay, so I'll hurry up. And okay, so when we do that in the late phase, we observe that we actually match the bifurcation model. Okay, so if we do, so this was validating a prediction of this model, but we can actually fit all two models to the trial-to-trial data, and we observe that indeed the unimodal model wins in the initial period, and then bimodal dynamics are there for the later period. And you can look at that in individual subjects. So the next interesting question is what happens in the passive condition, and especially these late activity, do they correspond to task-related activity? If they do, they should disappear in the passive condition. People are not doing a task on the stimulus. But if they correspond to conscious access, then they should remain maybe a lower intensity. So what happens? What prediction is validated? Well, none of them. So what we observe is that indeed in the passive condition, we still have late activity for these stimuli that are clearly above the threshold where you spontaneously become aware of the stimuli, up to 600 milliseconds after the stimulation, although you have nothing to do on the stimulus. But, okay, so you might be very dishonest, but you cannot say that this is a P300. So, so you have to admit that this is not a P300. So what happens? P300 has disappeared, but like late activity has not disappeared. One possible interpretation is that this activity is actually included in this activity. So here we might have core conscious access plus connecting that to executive area to make the task. And we can, so maybe I won't detail that. We can actually test this interpretation by doing cross-classification across the two conditions, passive or active. And we see that passive can decode active, but does not capture everything that's occurring in active. Active can decode passive and captures everything there is to see in the passive condition, validating the idea that passive is included in the active condition. And if we attempt a reconstruction of the sources, we can see that in this late phase for the active condition, we have a network of area, so a global workspace. For the passive condition, we also have a nice network of areas. And the difference between the two lies in the motor planning, motor cortex, okay? So if this is a global workspace, so the circulation and maintenance of an information for a special goal, this might be a global playground. So a circulation of some information in a network of areas, the maintenance of this information for no special agenda. Right. So the last question is, do we have bimodal dynamics or bifurcation dynamics in the passive session? So for these late activity, and the answer is yes, we validate our prediction of a burst of variability around threshold. And if we use the full model, we see that indeed the bifurcation model wins in a smaller time window between 200 and 500 milliseconds. And okay, so how much time do I have left? Still. So I still have 5 minutes? Okay, so, sorry? Okay. So, okay, perfect. So next, there are only two things I wanted to show you. These are very preliminary data. So as a follow-up of these initial studies, there are two things we wanted to do. First, investigate more precisely the neural underpinnings of these dynamics using intracranial electrodes. And the other one, obviously, is transfer this protocol into disorder consciousness patients. So first, a few preliminary data with intracranial recordings. So these are, same protocol that I did with Daphne Rimsky-Robert here. And so an electrode in Eschel's gyrus in the insula, in the inferior frontal, you see that they give nice activations to the different stimulation strengths. And if we look at our markers of dynamics in Eschel's gyrus here, where its first response, we see a nice increase in activity, in mean activity, flat variability, suggesting that we don't have bifurcation dynamics. Same for the late activity, negative activity here, nice decrease or nice increase in negative activity, flat variability, okay? In the insula, we see something different. We see that at late phases, we clearly have or mark bifurcation dynamics. And it's also very clear in the inferior frontal, starting from even earlier, 200, 300 milliseconds. And if we now just pick the trials with threshold simulation and sort the trials according to whether the subjects said that they heard the stimulus or not, we see that it makes no difference for Heschel's gyrus. And indeed, we don't see a bifurcation. There's no two groups of trials. But for the insula, initially, it seems to be similar activity up until 300 milliseconds for heard or not heard. But the late activity seems to split between these two types of trials. Right. So now the big question was, can we use these this new description of brain dynamics to investigate conscious access in DOC patients? So it's very preliminary again. So we transferred this protocol into patients, and what we're trying to do at the moment is really find patients that are on opposite ends of the spectrum to see whether we have completely different dynamics. I show you two examples. That's the example of a vegetative patient. That's the mean activity in response to increasing SNR, and you see that the SNRs are completely different. Here we go to 14 dB positive, so that corresponds to a normal conversation, and you see that there is a nice response of the brain to increasing stimulation, but variability of this response just scales with the mean, okay? And there is no burst at a specific threshold. Completely different patient, exit MCS, so presumably this person is regaining consciousness, and we know that from clinic. And here what we see first is that we missed the threshold, so that's bad. So we'll have to obviously improve the protocol to adapt to each patient. But still, we see that the profile of variability is completely different. It's not following the mean. We are saturating when we go above 10 dB. But still, around 0 dB, we might be around the threshold, and we seem to see a burst of variability at that level. Okay, lots of work to do, but I just wanted to give you a hint that just investigating these, with this very simple protocol, the dynamics of activity in these patients might give some interesting complementary information to the protocols that we already have in these patients. Okay, so to wrap up, after all these studies, I'm still more committed to the idea that there is a bifurcation between two ways of processing information in the wakeful brain, and it seems to be quite general. I'd like to also to make I think it also brings an interesting nuance in this notion of ignition that was developed by Stan Dehaene notably, that you can have nonlinear response to stimulation and still not bifurcation dynamics. So it's interesting to see this separation. And so I'm suggesting that maybe bifurcation dynamics might be an interesting signature of conscious access, even in the absence of report, and that we might want to use them to assess conscious perception in patients or in different states of consciousness. And finally, I'm very glad that to see also, thanks to intense debates with notably Victor Lame and now to go to Shia about the fact that maybe what we're studying is only task related. Now we've we've really reached the time when everybody embraces the idea that while we've been studying brain activity related to subjective report, now we can try to do the return path and look at brain activity and try to infer subjective experience from that. And lastly, I'd like to thank my current team and my collaborators, and thank you for your attention. Okay.
Hi, from Israel. This recording in the intracranial that you did and you called the inferior frontal location, is it part of the auditory system, language system? Was it on the left side, on the right side, or was it more frontal, decision?
Okay, so it's on the left. But I should go back to the precise anatomy. I can answer you by email, but not just now. So you think it might be in Broca's area? That's your hint.
I think it might be a high order auditory area, and then it fits very nicely with the visual suggestion that this bifurcation or ignition happens in high order visual areas. So I'm interested if there is a parallel between the auditory and the visual in this respect.
Yeah. Okay. So obviously, I should check. It would also be interesting to see the correspondence with the response and whether we have a response that is time locked to the button press to really see whether it falls rather on the perceptual or on the task-related components. But I still need to investigate that.
Hi, Claire, this is Victor.
Hello, Victor.
Hello. I really like this bifurcation dynamics analysis you do. So what you show is that in the absence of report, you still get the bifurcation dynamics, but now limited to, say, well, the back of the head, sort of, or middle of the head, what have you. The global playground, as you say. Would you expect this bifurcation dynamics to also happen, say, in cases of inattention, or if people are not attending to these stimuli at all?
Okay, so during inattention, that's what you're saying. So, for example, if I really completely diverted the attention of my participant from what they're listening to.
Yeah.
Yes?
Yeah, that would be the question.
Yes.
Yeah.
Okay. So my prediction would be that bifurcation dynamics would be abolished in the same way that the late activity is abolished in the attentional blink when people fail to see the second target. But that's my prediction. And in relation to your work, I think that the nonlinearities we see in the initial activity might very well relate to sensory recurrence in within the auditory cortex.
Okay, but you would need attention to have these bifurcation dynamics. So you would not say it's an intrinsic property, say, of the sensory cortex to have these bifurcation dynamics.
Well, at the moment, I'm still committed to the global workspace idea that it seems to arise at the brain scale. I mean, it still arises around 300 milliseconds, as we've seen previously. And I'm still committed to the idea that these bifurcations correspond to the ignition of the global workspace, as opposed to local recurrence. But this needs to be investigated, obviously.
Thank you.
Thank you, Claire.
Thank you.
Do you want to...
Okay.
All right, so we have our last speaker of the day. It's going to be Peter Rodsema with a talk on, let me find your title, visual stimuli that do and do not mask make into consciousness. Where is
I don't hear.
I'm too old to hear it.
If I wonder, do you know where should I ask Mike?
No, I think I know. I think I know what the problem is.
Yes, that was it.
It's amazing because I don't hear anything.
No, really? It was pretty annoying. Okay, so our last speaker, Peter Robson.
Okay, so I would like to thank the organizers for putting together this fantastic symposium. So I would like to discuss what a visual stimulus needs to do to reach conscious awareness. And I'll start with a very brief introduction. You know all this stuff, right? So this is the macaque visual system. I'm going to present quite some data from the macaque monkey. All these different colors refer to areas involved in vision. So this is the famous hierarchical scheme from Van Essen. If you present a new stimulus, it enters in the cortex in V1 and is then propagated to higher hierarchical levels. Victor talked about it this morning, feed-forward processing. That gives rise to the detection of very simple features in low-level areas, feature constellations of intermediate complexity in intermediate areas, and more complex object categories in high-level areas. Okay, but there's also this recurrent processing enabled by lateral connections and also feedback connections, allowing for recirculation of the activity. So as Victor talked about this morning, that's important for perceptual organization and also attentional processes. But of course, we're talking about awareness. So what is the relation to awareness? So that's what I want to talk about today. I'm first going to give a brief introduction on the relationship between feed-forward and feedback processing on awareness. Then I'm going to test these ideas in three different tasks. A very simple task, and then a more complicated task, basically what Victor talked about, and then an even more complicated task. See what we learn. Okay? So this is the idea that Victor also presented this morning. So you present the visual stimulus, then there is basically a wave of feed-forward processing that propagates the information to higher cortical areas. Might be unconscious. But then you have this reciprocal interactions where higher areas also start to influence neurons at lower hierarchical levels. And in this paper that we indeed published 20 years ago, we speculated, and then we were really focused actually on the posterior part of the cortex, that this is important for conscious awareness. Now Victor wrote most of these parts. I kind of thought, let consciousness, let Victor deal with this, I'll deal with attention. Okay, now I'm here, so I have to deal with it. Okay, so this is what we speculated. Now around the same time, maybe even a bit earlier, Stan de Hane published similar papers also focusing on the reciprocal interactions, and he has this process called cognition. I'm going to talk about more about that today. So these are some of the papers. He was more focused on the frontal and prefrontal cortex, but some expressing quite related ideas. Okay? So, and ignition, I thought, is an interesting concept. So the idea of ignition is that if it happens, then if you take away the stimulus, there's sort of persistent activity. So a subject will be able to report about a stimulus that's no longer there. That's, I think, also implicit in this concept of ignition. Okay, and then according to Stan, if a stimulus is weak, sometimes it will fail to cause ignition, and then it will not be reported, and sometimes this recurrent activity can occur, and then it will be reported. So actually, I now think of it as ignition as the ability of a weak stimulus to kind of enter into working memory, and I'm going to show some evidence in favor of this idea. Okay, so let's go to a very simple task, the detection of contrast. And so I'm going to ask you to kind of participate in this simple task. So I'm going to present a stimulus about here. If you see it, please raise your hand. If you don't see it, don't raise your hand. So let's do this. 1, 2, 3. Okay, that was easy. Now this is for the very good viewers. I'm going to make it more difficult. 1, 2, 3. Who saw it? One person. Okay, maybe a bit less difficult. 1, 2, 3. Yeah. Last chance. 1, 2, 3. Okay, honestly, there was only a stimulus on the first and on the third trial. Those who raised their hand on the second and the fourth, this is what we call false alarms. Nothing to worry about. You don't have to go to Mike Shetland. Okay, so these phenomena are very well described by signal detection theory. And in signal detection theory, the idea is that if there's a stimulus, if say it's a weak stimulus, you get sort of a distribution, and it's called the internal representation of signal strength that has some stochasticity. So on the trial with the stimulus, you will be sampling from this distribution. Say it will be this internal representation of signal strength. Now you have to decide, am I going to say yes or no? So for that, the signal detection theory invokes a threshold. If the stimulus is stronger than the threshold, you're going to say yes, it will be a hit. Occasionally it will stay below the threshold, and because of the stochasticity, so that would be a miss. If there's no stimulus, you will be sampling from the other distribution with a lower mean. So typically it will stay below the threshold. You'll say no, it's a correct rejection, and sometimes it will just by the stochasticity go across the threshold, and you say yes, it will be a false alarm. Okay, now what is interesting is to think what would happen if you present a stronger stimulus. If you keep the same threshold, you're still going to have quite a number of false alarms, so you might want to raise your threshold. You won't have many false alarms, and you will not have a large increase in the number of misses. Because if you have this high threshold, you'll be do quite poorly for a weak stimulus, because now you're going to have quite a number of misses. So this is what signal detection theory is about, is kind of setting the threshold, and it's quite able to account for a lot of human psychophysical data. Now one thing that we were interested in is, where is this internal representation of signal strength in the brain? Is it somewhere? And also, what causes this variability? What causes the width of this distribution across trials? So we trained monkeys on the very same task you've been doing. So they were looking at the fixation point. In 50% of the trials we presented a stimulus of varying contrast. On 50% of the trials we presented nothing. If there was a stimulus, the monkeys were supposed to make an eye movement to that location where it previously was. And if they didn't see anything, they were supposed to make an eye movement to this, what we call reject dot, that was always there, allowing the animals to report that they didn't see anything. Okay? Then we varied the contrast of the stimulus using a staircase procedure. And basically, if the contrast was high, they always saw it. If it was low, they never or hardly ever saw it. There were some false alarms. We defined two thresholds that was just convenient for data analysis. A high threshold where the animals reached 80% or more, and a low threshold where their accuracy was 40%. If it was higher than the high threshold, we called the trials an easy one. If it was below the low threshold, we called it a difficult one. And in between the thresholds, we said this is a trial of intermediate difficulty. And then we recorded from V1, V4, and dorsolateral prefrontal cortex. And when we recorded from V1, I was in for a surprise. So what we're showing here is stimuli of the same contrast, but some of them were reported by the animal and some of them were not. Okay, so even at the level of V1, you see a difference, as if there is variability in the fidelity of propagation from the retina to cell in the visual cortex. I wasn't expecting that. And you see that this difference is also there for the intermediate stimuli, and for the easy stimuli, at least proportionally, the difference is smaller. And what is also interesting is to know that the easy miss stimuli actually have more activity than the difficult hit stimuli. So a certain level of activity in V1 is not a guarantee that you will enter consciousness. Information can get lost downstream from area V1, okay, at maybe going up to the level where it's consciously perceived. Okay, so we can then look at the ratio between the activity on the trials where the monkeys didn't see it and those that they did. So we call that a misfraction. You see it increases, basically the ratio becomes larger. Okay, so in V4 we basically got the same result. Also here, easy mistrials elicit more activity than difficult hit trials. So also a certain activity level in V4 is no guarantee that the stimulus will enter consciousness. Okay, in dorsolateral prefrontal cortex we got this sort of ignition-like profile. Now, I should warn you, here we're recording from an eye movement region. So we're not dissociating the monkey's eye movement plan from phenomenal awareness. And there we see this threshold phenomenon. So whenever the animal is going to report, you get a high activity, and whenever he misses it, you get a low activity. You see there is a little bit of activity here on the easy trials, but apparently not enough to reach the threshold for this self-sustained activity. So misfraction is always low here. And here you could have this, you could put the threshold. If activity reaches this threshold, the animal is going to report it. Now we also looked at false alarms, so there was no stimulus, or correct rejections. If there was a correct rejection, activity was always low, there was nothing in the receptive field. If there was a false alarm, you get this ramping of activity, and we actually believe what's going on here is that, we can't prove it, but on some trials, actually there's no stimulus, so there's no natural time zero, but sometimes maybe there's a spontaneous ignition, activity goes up to a high state. If this happens at different time points for different trials, even in average across trials, you get this slow ramping, but we can't prove that this is the case. Okay, so if there is any region that is reminiscent of this signal detection theory, then it's going to be dorsolateral prefrontal cortex. Okay, so I showed these data to Stan, and he said, "Oh, I can model that. I've modeled things like that." Okay, so and then he made a model that has only 5 neurons, 5 stochastic neurons. The whole brain is modeled by 5 neurons. There's a neuron in the LGN, and then the activity gets propagated to high areas. There are feed-forward connections, there are self-connections, and there are feedback connections. And he made the connections between parietal and frontal cortex and the self-connections here so strong that if activity reaches a certain level, it can self-sustain itself. Okay? So that would be ignition. So now even if you take away the stimulus, the activity will persist. It now basically transformed into a working memory trace. Okay, that would be a hit trial. Because of the stochasticity, the same stimulus might, on some trials, not reach this critical threshold for self-sustained activity. So if you then take the stimulus away, activity subsides again. That would be a miss. Okay, this is then model V1 and model V4 and model frontal cortex. These are the correct rejections and these are the false alarms. So you go. Now we can prove that this slow ramping is actually due to the stochastic neurons sometimes spontaneously crossing the threshold for ignition and then causing self-sustained activity. That happens at different time points in different trials. In the model you can see that. So this is the model. It's a bit embarrassing. I think Stan did it on a Sunday afternoon. This is the data, and we worked on it very hard for many years. Okay, and you see there's quite a nice correspondence. So it's pretty good description of the data. So what I take from this is there's variation in the efficiency of feed-forward processing that then explains the variation in this internal signal strength across trials. The threshold for perception, the threshold of the signal detection theory could correspond to the threshold for ignition, the threshold for the self-sustained activity. And D prime could be a measure for the quality of information transfer up to the level that is kind of going to make this bifurcation. Okay? So, Victor and I said it's going down here, but actually the data that I've presented so far is quite compatible with the critical components being mainly in the frontal cortex. V1 seems to only be a relay of the information to higher visual areas. But that is in a simple task. Okay, now we're going to go to a more complicated task. This is the task that Victor was talking about this morning. So I'm going to give you a little bit more background here. So if you see a picture like this, of course you see a lot of information. You probably don't have a hard time seeing that this and this is part of the same zebra. Now that's quite remarkable because vision starts with fragmentation. Receptive fields in V1 are very small, and at first sight they only seem to be caring about the information in a very small fragment of the visual space. This is not how we see, right? And actually we think what's going on is there are these grouping operations that take place, and if you're interested in the central zebra, there is this labeling process, labeling all these image elements that belong to a single zebra with enhanced activity. Okay? So this is what the brain makes of it. And actually I would like to kind of defend the idea that this labeling process is also very important for visual awareness. Okay, so this is the test that Victor was talking about. So one example where you need to do perceptual organization. So everybody sees a square figure composed of line elements of one orientation on the background with line elements of the opposite orientation. So this is what Victor showed 25 years ago, that you get more activity if image elements that are identical in two situations belong to a figure as opposed to a background. That's what you see here. And this is from a study also many years ago where the link with conscious awareness was established. I think it's a really nice paper. So this was done by Hans Super. So the monkey saw a figure ground stimulus and he was supposed to make an eye movement to the center of the figure. And if the receptive field can be on the figure or on the background, and on those trials where the animal perceived it, there is this figure ground modulation, more activity if the receptive field is on the figure. But on those trials where the animal failed to see it, Victor presented it this morning, there's not this effect. Now I would like to believe that the animals probably saw all these image elements, but on those particular trials there was no figure ground modulation. He probably failed to have the perceptual organization in place. Okay? So maybe there are conditions where you also need these reciprocal interactions with the visual cortex. But we never had causal evidence for that. So now I want to present causal evidence. So this is figure ground segregation in the monkey, figure ground. You can also see it in other species. We had this one lucky case with a patient with epilepsy whose epilepsy always started with visual hallucinations. So we could get multi-unit activity from V3. We also got figure ground modulation. But now, and that is very convenient, we also can see it in a mouse. Okay? So more activity listed by a figure than a background. But that's very useful because in the mouse we have very powerful methods to manipulate brain activity. So this is the task that we have the mouse doing. So he looks at the figure ground stimulus. If he sees a figure on the left, he licks to the left. If he sees a figure on the right, he licks to the right. You have contrast-defined figures, no figure ground segregation required, orientation-defined, phase-defined, and texture-defined. This is how close we can approximate the monkey stimuli. And they can do it. I mean, they're not stellar, they're mice, and they never reach 90 or 100% for the figure ground stimuli, but they're clearly above chance level. We have to work with this. Okay, and then we can do the recording. So this is a single multi-unit activity recording. So this is when there's a contrast stimulus as opposed to a gray screen. So this is visually driven activity. This is figure ground modulation. So this is figure, and in the background you get less activity. And this is for orientation defined. This is phase defined. It happens a bit later. Actually, you can compute the latency by subtracting the background response from the figure response and then fit the curve. So it's for orientation defined figures it's 95 milliseconds, and for phase defined figures it's a little bit later. Okay. And now the dream experiment. We always wanted to do that, but we never got it to work. But now we get it to work in mice. Okay, so we want to do silencing. And what we're doing here is we use this very powerful optogenetic silencer. It's called GT-ACR2. We inject it in primary visual cortex. And then we shine a light. We can block the entire visual activity in V1. Abolish all the spikes. Or we can just let through the feed-forward response and only abolish the delayed activity. Or let through a little bit of the delayed activity, and so on and so forth. Okay, that's what we're going to do. Okay, so this is the contrast task. So there's no figure ground segregation required. And what you see here is the individual mice. The green one is the average across mice. You see that they have about 85% correct if we don't shine a light. And if we shine a light early on, abolishing the entire visually driven activity, you see they don't reach chance level. They're still about 60% correct. Maybe it's blindsight in mice because we've knocked out V1. Who knows? You see that they recover after about 60 milliseconds, and after about 80 milliseconds they're at ceiling performance. So, if we just let through the feed-forward response, the animals can do the task. So in this task, V1 seems to act as simply a relay station, sending the information to higher areas. But in the figure ground case, it was different. So here, when we silence V1 early on, the animals are completely at chance level. And we have to postpone the light to say about 120 milliseconds before they're back to ceiling performance. Okay, so they do need a little bit of this late activity to perceive the stimulus. And in the phase-defined figures, it's even later. It's maybe about 150 milliseconds before they reach ceiling performance. Okay, so we are excited about this. So if it's contrast detection, V1 seems to act as a relay, but if it's about perceptual organization, you really need the late V1 activity. Okay, so that's the idea. So maybe this delayed activity in some cases is really related to your ability to see that particular type of perceptual organization. Okay, so let me summarize this. So activity gets propagated to higher areas. Maybe this is the time that you realize there's something on the screen, there are texture elements, but they're not yet organized. For that you need the feedback. So then these image elements become labeled with enhanced activity. And then you can report also about the perceptual organization, and maybe also report that it's a square-like figure. Okay? So in contrast detection, this seems to be the case, but in perceptual organization, you need also a link to lower visual areas. Okay, so in the absence of a visual stimulus, my kind of feeling about it is that what is in the awareness corresponds quite well to what is in your working memory. I guess many people will not agree, because I know consciousness is a hopelessly divided field. Maybe some people agree with me. In the presence of a visual stimulus, what is in awareness corresponds to what is in working memory, plus the items that are labeled with enhanced activity in the visual cortex. Okay, object-based attention. Now let's take this one step further. So now I'm going to talk about a task that requires more perceptual organization. So again, I would like you to participate in this task. So what I'm going to do, I'm going to present two curves. And one of these curves is going to make a connection between the red dot and either the green dot or the blue dot. If you think it's the green dot that's connected, you want to raise your left hand. And if you think it's the blue dot, you want to raise your right hand. Maybe you want to be a bit faster than your neighbor. Who knows? Okay, you ready for this? 1, 2, 3. Yeah, I see mostly left hands. It takes a bit of time. Okay, maybe you want to take revenge. Now we start from this side. 1, 2, 3. It takes a lot of time. So you can spend maybe, on average, I think it's about 10 seconds before you see it. It's remarkable, right? It's a stationary stimulus. All the information is there from time 1. But you need 10 seconds. This is recurrent processing. Okay, what you do here is you probably would move your eyes here where you start. And then you're going to make a sequence of eye movements along the curve that you're mentally tracing, at some point arriving at the blue dot. Now this is a stationary stimulus, so you're only going to make saccadic eye movements. So if your eye is here, you're probably mentally tracing this curve for a stretch, and then you're making an eye movement. Then you trace ahead, make an eye movement, trace ahead, make an eye movement. So of the 10 seconds that you're processing the stimulus, it's only the 1 second, but 10% is eye movement time. Your eyes kind of are flying around. 9 seconds is pure mental work. Okay? So, how does this work in the visual cortex? Well, we train monkeys on a task that requires this. They look at a fixation point, then they see two curves. One of them kind of makes the connection between the fixation point and a larger red circle. This is the target curve, and the target circle, this one is not connected, so it's a distractor. And then after a delay, we remove the fixation point. Now the animal is supposed to make an eye movement. We're always recording from the phase that the animal is only thinking about the curves, not yet moving the eyes. So here V1 receptive field, here it's on the target curve, here it's on the distractor. Basically get precisely the same effect that we saw on the figure ground. So if the receptive field, if something enters into the receptive field here and here, it's the same. You get a visually driven response that does not discriminate, because what is in the receptive field is identical. But then the monkey starts to mentally trace, and the moment he reaches the receptive field, you get a boost of the activity level. Okay, and also if the two curves intersect each other. Now this is an interesting case in which we recorded from two groups of neurons at the same time. Your receptive field 1 is close to where the animal starts to trace, and receptive field 2 is farther along the curve. And Well, the first thing you see, the visual response, is the same for the two recording sites. It's just a delay between the cell in the retina and cell in the visual cortex. But you see that this response modulation happens early at receptive field 1, but it's close to where the animal starts to trace. Then at receptive field 2, that is much further along the curve. And we can account for these type of data by what we call a growth cone model of object-based attention, where activity flows along the curve, but with a speed that depends on the distance between the curves. So if curves are very far apart, you can probably use large receptive fields and cross a lot of space in only a single synapse. If curves are nearby, you don't want to use these large receptive fields because activity would spread from one curve into the other curve. So now you need to use small receptive fields, but then you will go slower because you need many more steps to cross the same amount of visual space. Okay, and we can then estimate the number of required growth cone shifts. We say that the growth cone is centered on the target curve and just does not touch the distractor. So this would be about 1 growth cone away from the fixation point, this one about 3 growth cone away from the fixation point. So this is the number of required growth cone shifts, and this is the latency of this response modulation. And we get a regression of about 50 milliseconds per receptive field, which is quite slow. And maybe explaining why in this stats that I presented to you, you really, these delays are going to add up and reach the many second range. Okay? So let me summarize this for you. The next time you enter into the kitchen, you want to switch on the hairdryer. What are you going to do? Well, we now know there's a very simple algorithm implemented in our brain that solves this problem. And it might start in an area with relatively large receptive fields, but now the curves are too close together for a V4 receptive field. Fortunately, there are feedback connections, so in V1 is also informed about the tremendous progress that is being made in V4. V1 might take over, it will go a bit slower, and then at some point V1 will inform V2 and V2 and V4, and they can then take over again. That's how we think about it. Okay, this is neurophysiology. In psychology, we did quite a number of studies in human participants, and this corresponds quite well to what psychologists call object-based attention. So there's quite a nice match between what psychologists describe as object-based attention and what we see in the visual cortex as a propagation of enhanced neural activity. Okay? Now summarize this as well, now in the context of consciousness theories. So what's going on here? So suppose you want to start to trace a curve that starts with a red marker. Well, you might want to start a visual search for a red marker in the display. Okay, and we know actually that there are feedback connections to early visual areas that are going to highlight that location in the visual field where the color resides that you're looking for. Okay, then you might want to make an eye movement to that red dot. And now, I think that happens in working memory. Now the trace operator, that's maybe another set of neurons that are implementing this operator, are going to be activated in working memory. And we see that also works on the visual cortex, labeling the curve with enhanced activity. Okay, and so now you kind of identify the green circle, the one that's associated with the left-hand response. You might also now have activated some of these higher level shape features. So this is a part of the curve that has a particular shape. So this could also now be part of your working memory. If you take the stimulus away, you might still remember a little bit about this shape. Okay, so I think that in working memory are those image elements that are on your mind, that you're thinking about, that are part of your task set. So, and of course the active mental program in my example, searching for something, tracing a curve. They interact with the visual representation. So actually this is something that is also part of the global neural workspace theory. It's this kind of broadcasting of information. Broadcasting of information for me is the same as object-based attention, and the interaction between working memory representations and the visual representations. That's why we perceive multi-feature, multimodal objects, because of this process. Okay? These working memories also activate the motor system, for instance the eye movement or your hand movement, as a result of this processing. And of course they also change. So the set of active working memories as you're doing the task is changing while you're performing the task. And of course the challenge is to understand how you go from one state in a cognitive problem to the other state in a cognitive problem through a number of intermediate steps that correspond to different states of your working memory. Okay, so this is kind of the bottom line, how I now think about it. In the beginning I didn't want to say too much about consciousness, but now I'm here, 20 years later, I'm forced to talk about it. So for me, what is in awareness corresponds quite well to what is in working memory, plus the items in the visual field that are labeled with object-based attention. And about this idea of a sequence of cognitive states, actually we wrote a paper quite a number of years ago, what we call the human Turing machine, that expresses many of these ideas. Okay? So this is the thing that is in your awareness. It corresponds quite well to what is labeled with object-based attention. Okay, and I would like to mention that some of these ideas that are now seem to be possible to reconcile some of these ideas will come out in a paper that I think will be published in March in Neuron. These are the people who did the work. So the early work was done together with Victor, Henk Spekreis, and Arasu Ismaili. These are the people, including Stan, who did the modeling, who did the work on the stimuli that have a very weak contrast. And this is the whole group of people from the Netherlands Institute of Neuroscience that were involved in the mouse optogenetics project. Thank you very much.
We have number. Okay. Be loud.
Loud? Okay. Comment and a question. At the first part of your talk, the model, I thought it was a failure, not a success, because you had persistent activity in the late stage, even on the misses.
No.
The model doesn't predict it, but it's in the data.
Let's go there. Let's go there fast. So the model and the data next to each other.
It's in the red curves.
So in the data, where do you mean this
Yeah.
activity? Yeah, that's what's also in the model, right? So that is kind of a sort of there is probably some resonance between the frontal cortex and the parietal cortex, but not enough to really cause this going through the bifurcation and then cause persistent activity. So it basically dies down. That's what you see here, and it's also what you see here.
I see persistent activity at a lower level. I would say
Here,
Yeah.
but it goes here, it goes away, right?
Look at the middle one.
Here, this one?
Yeah.
Okay.
Okay. Anyway, so I think that's
Probably not enough for the animal to report, it seems.
apparently not.
No.
But enough to make some kind of ignition to have persistent activity.
Um,
And let me ask you the second question, if you don't mind. We can talk about it later. But so the tracing, I understand the idea, and I think it's a beautiful idea of using feedback in order to sort of tag the source of information. But you still need some element downstream that decides on connectedness. In fact, it's making a sequence of decisions depending on the size of your system. So what's the missing mechanism there?
So some of these mechanisms are actually embedded in the visual cortex. So Victor showed in his talk on the connectivity. So in V1, if you have two contour elements that are well aligned, activity will tend to propagate from here to here, just because of the connectivity in V1. I think Rafi even had the paper on that, or on related connectivity data in monkeys. Now, it's not only necessary, or not the only way to go from here to here in V1, it's just horizontal connections. It could also go up the hierarchy to V2 and V4 and then come down. So we don't have strong tools to dissociate between those two possibilities. But a lot of this connectivity that allows activity to spread according to what are the Gestalt rules of good continuation or common fate and so on and so forth, I think these are embedded in the wires.
Well, while this is on, of course, these signal detection theory models have always been, well, I would say rejected in the context of consciousness, because, of course, you can lower or heighten your decision criterion. For example, if I would put a gun to your head and say I will shoot you if ever you give a false alarm, well, there will be no hits, I assume. But of course, it doesn't assume that that doesn't imply that then you don't have any conscious sensations anymore of the stimuli that you're seeing, even though you give 0 hits. So, and of course, this has been, people have tried to adopt this with, let's say, 2-stage models, or so that wouldn't that better fit maybe even all the data? A model where you have a first stage where you have, say, like a bifurcation thing happening, like in the global playground, you might say, if we refer to the talk that we just heard, And then the second one where you have this decision stage, which is then operating on that first stage.
Yes, I agree. So I would say yes. And what we do in here, we're recording from the basically frontal eye field, right? So the area where the animal has planning is the eye movement, which is the response required from the animal. So it would be, the next step would be testing an animal and not telling him beforehand whether he's supposed to make an eye movement or a hand movement, right? So then maybe you won't even see it in the frontal cortex, but I bet there are other regions in the frontal cortex that code the plan to do something with the stimulus in a response modality independent manner, although might have to look for those. Now what you're saying, suppose that on a certain number of trials it would be really bad for you if you have a false alarm, so that might also then influence these later stages of kind of the percept or the working memory of something that you saw and then connecting it to a motor response.
Would you say it would influence also the, well, let's say the activity in the visual cortex if I put this gun to your head?
That's a good question. I don't have a strong opinion on it.
Okay.
Yeah.
You showed the tasks with increasing difficulty, and when you're comparing a mouse and monkey and man and humans with respect to these tasks, do you see any differences in terms of this kind of data? Not necessarily qualitative differences, but quantitative differences. And if you do you expect that there will be some kind of ecological adaptations, that some species would be very good at a particular task and it will come to awareness very quickly, while others would not? I mean,
there are many differences between species, obviously. In our hands, the most visible one is the ability to train. So the mice, it took us forever. We spent 2 years on finding a paradigm where we had the mice more or less reliably reporting what they saw. It's probably our inability to find a more ecological valid approach to this. The monkeys we train, well, in this task it doesn't require a lot of training, maybe a week or 2 weeks, and they can already do it. And a human, you just sit in front of a monitor and you say, oh, well, this is, you're going to see a figure, make an eye movement there, right? So there are huge differences, which is probably related to the amount of frontal cortex, the ability of humans to understand the instructions, and their ability to take instructions and translate it into a mental program that can be readily executed. I think that's a really interesting question.
Following on Victor's comment, I mean, typically when we are doing a threshold experiment, we expect the effect of seen, not seen to be in the weak stimulus, not in the strong stimulus. Now I look at your data and I see that in V4, for the weak stimulus, there is a sort of linear, non-linear activation for seen versus miss, and for the strong stimulus, it seems to disappear, which is very puzzling. So I have a very simple common sense question. If you actually look at the strong stimulus, do you always see it, or it also sometimes disappears? Just for assuming that your vision is a bit like the monkey, and you actually look at the strong stimulus. To me, it seems that the monkey decided, maybe because of criterion or other things, that he actually subjectively saw the stimulus, but decided not to report, and you call it a miss. But the weak stimulus is the place where we are expecting this kind of changes to happen.
Yeah, your question is about where is phenomenal experience, right? And this paradigm does not allow us to tell it. And it's related to Victor's question, so if you put a gun to my head and I'm going to shoot you if you have a false alarm.
But how do you explain the weak effect?
Well, it could be that indeed the animal failed. I mean, we cannot ask the animal, but if a human would sit there and have this amount of V4 activity, he could still, oh, I didn't see it. I don't know, it could come out. In the figure ground situation, I think that's also an interesting case, right? So there, you obviously saw, or I think the monkey saw the texture elements, but probably failed to organize it. Now what happens here? I can only speculate. So it could be that they would report, I didn't see anything, or it could be that they were just not motivated to press the button, and that would also be kind of a miss, of course.
I was wondering whether you were looking also like on something like LGN, despite it is a non-cortical area.
Yes, so we haven't looked there. Of course, it would be interesting to look at many structures, not only V1, V4, and frontal eye fields. As we are seeing a difference already in the feed-forward response in V1, it would maybe not be terribly surprising if that there's even some of this effect there in the LGN. But right now it's speculation, right? We would have to put an electrode there to really find out.
I have two questions. One, in your model, at some point there was LGN, and I don't know what happened to it later on in the story. It disappeared. Is it still there, or no?
Oh, it was not plotted here, but so LGN would have been here.
So why not feedback to LGN?
Oh, you could. Oh, sure.
So
yeah.
Is there evidence? I mean,
yeah, there is evidence for it. There are feedback effects on the LGN.
So there is a trace for awareness in LGN also.
That I don't know. I mean, that was also her question. So I wouldn't be surprised if this very early effect that we see in V1, this one, that some of it is visible in the LGN, because we're just looking at the not so reliable connection between the eye and the brain, I suppose. So maybe some of that variability is contributed by the stochasticity of the LGN neurons.
So a tracing model will be powerful enough to tell you whether you want to trace it back to the LGN or to the retina, or is there any functional criterion?
No.
Predictive?
No.
Okay.
No, I think that even in the retina cells are stochastic, right? In the LGN cells are stochastic.
Sure.
So maybe you're seeing the accumulation of these stochastic effects that sometimes may get amplified because of the feed-forward and feedback connections, right? So that I suppose that amplifies these stochastic effects.
And related to it, so in the experimental results, when you see these two traces in the weak in V1, are they two traces, or it's really continuum that you group?
Yeah, these are averages, of course. So if you look at single trials, there's a lot of variability, and as we see already in the data, a certain level of activity in V1 is no guarantee. Something can go wrong downstream due to the stochasticity that is downstream.
Just wanted to say the result with the inactivation of V1 in the mouse is beautiful, but I don't see why it picks out the feedback modulation. If the animal needs time to solve figure ground, then maybe it just can't see.
Yes, but it needs apparently V1, or at least that's the area that we knock out. So that's the one area where we're really sure that we're knocking out all the spikes. Now, it can be that by doing this manipulation, there are also indirect effects in another area that we don't control, but that might be affected by the fact that we silenced V1. But these areas were apparently not important in the contrast task. So much we can say, because there, if we silence the late activity in V1, nothing happened.
But if you shut off the stimulus early, and the animal still has a problem, then you'd say, well, you just need V1, not the feedback. Or in a very fancy experiment, you express all over the visual cortex, but in retrograde viruses or something, and you only shut down the feedback. Now if you get your result, then you've...
That would be a more elegant experiment. So I think this is already telling, right? So apparently you need these late V1 spikes to be able to do the task, which is more than we ever hoped for to find. And in monkey we couldn't get optogenetics to work, at least not as reliably. Now the next beautiful experiment would be to kind of only shut down the feedback connections, right? Like expressing the absence in the feedback connections. We tried that a little bit. It's not so easy. So this GT-ACR2 is a very powerful silencer, and there are some other powerful silencers around. But their effects on axons, they're not always as reliable. So I think the idea is that in axons also maybe the chloride reversal potential is a bit different. So if you shut shining light on the axons with the GT-ACR2, actually they excite them instead of inhibiting them. So it's a really cool experiment, but maybe in 20 years then we'll be able to do it.
Thank you, Peter. So this is it after 2.5 days of intense discussions. Supposedly we had concluding remarks, which none of us really planned for, but since I was lazy, because I went to have the chair of this session because it was short, what I did not count is that I had to work hard to give the concluding remarks, which I will spare you in the long run. But what I do want to share with you is a couple of thoughts that came out through this 5 months that we had been dancing around the question, and also like my strong disappointment in 2012 when we put out this paper on the neural correlate of consciousness that really led me to quit my research on consciousness because I thought this is useless and we'll never find the answer, and how stimulating has been these 5 months plus the meeting that we had today, because maybe that program was wrong, but I think it had pushed us to think of better questions. So maybe the way in which we were approaching the question was the wrong one, but definitely not the question. And I want to conclude by in the morning we had a discussion with Renata, and she was, thinking more from the younger generation, like what does it feel for somebody who is just coming into, like doing research on consciousness? And definitely when I came, it was this exciting moment, when, Rafi was, pushing out research, Stan was pushing out research, Julio had this idea of the photodiode, which I thought was very weird, but which, they were considering. And it really felt like we're going to find the answer, right? And we are, and now what is cool is that we have 20 years after, we have review papers, and the answers turn out to be far more complicated, right? So we don't really know, or we became far more aware as to what were the problems and the limitations of our methods on the one hand. But also I think that what is interesting is that what has entered into the research that we do now that wasn't there before was to really the real challenge, which was how are we going to get to the qualia aspect. So when I started, it was like, oh, let's find for on and off, which is, it's an interesting question. I was pushing you yesterday. It's like, okay, once we find this common principle, it's going to be really cool. Yet, what I really want to know is why when I hear, it feels different than when I see. So like the first part is interesting, I have no doubt, but the second one is the real challenge, right? So what I feel is that at least where we have made progress is in daring to actually ask that question. Definitely we don't have answers to that, but at least we are daring to ask it. And I think that these, at least, what I feel is it's re-energizing, at least for myself, is that we now are going to try to develop different methods, geometries, whatever. We may not really know the answer right now, but at least we are daring to face it, right? I think in that sense there has been progress. At least like let's just face that the question was not just where we're on or off. Right? That's just one part. The second aspect I want to share that also across that trajectory of thinking that there's a lot of distillation in the process, that things that were having nothing to do with consciousness. One of those was memory. And these 5 months have been this, like, it's not just these 5 months, like it has been in my mind forever, like questions of temporality. Maybe memory doesn't have anything to do with my snapshot moment, but I have a sense of continuity, and maybe memories actually do play a role. So, there's also, being honest that maybe the questions that we were asking before were just too simplistic. So we have to really take the challenge of memory maybe does play a role, right? And if it's memories, what kind of memories, right? And maybe it plays a role the other way around. Maybe memories also, maybe consciousness also allows for certain memories to start that. I think that we're just asking the right questions, like by not simplifying everything. And taking that challenge as well, Liad, Peter, and a number of us have been, working really hard on, at least my quest is like, I know that we're all wrong, that I can tell you, but what I'm really interested in, like, who is more wrong than others, And so like the Templeton Foundation came up with this idea of let's do adversarial collaboration. And I think it's interesting for many sociological aspects, on the one hand, because one needs to be brave enough to say, okay, look, these are my predictions and I am willing to stand by them and not change them post-hoc, which is something that I have seen across the years. So at least it gives us a chance to say, okay, these are my predictions, I freeze them, and I will be brave enough to, then either make a public committal that I had to change my mind, or just, dismiss my theory. So I think it's interesting, on the one hand, it's going to make us to hopefully at least advance in terms of just cleaning up the theoretical landscape. The second, and the thing that at least to me it's much more interesting, is we're going to have data sets with monkeys, hopefully, with mice, with humans, that are going to be available for the whole scientific community. And at least to me, like, this is even more important than knowing, who's right and who's wrong. Now everyone, from, Chile where I was born, to Germany, to here, to everywhere, will be able to look at those data. And this is something that we need to think more as a community, like how to do science in a more socialistic way. Because I happen to actually be in the sense of, like sharing, right? I was born in Chile where, I didn't have the chance, and I will never have been able to, do the research that I do now, just because I don't have the means. But if the data are out there, if we all put our data out there, we could actually do much more. So it's also an invitation to say, like, look, let's just share our ideas, but also let's share our data, so that we can, test those ideas. And the last one, I hope that this was an invitation for you to think harder about consciousness and come to Tel Aviv in June for the conference that they are running here, and maybe get some nuances. So thank you. And there is pizza and wine, I think, downstairs.